Okay, I want to give you a kind of a video diagram of what we would do in the lab for the dilution series. A dilution series is a combination of two things. The dilution is referring to the fact that we start off with a rather concentrated culture of cells. So I could draw them in here as dots. So there would be so many cells in here that obviously you wouldn't see individual cells in there, but if we were to put any amount of this liquid onto a petri dish and let those grow up, there would just be so many cells that they wouldn't grow up into colonies that we could distinguish from each other. So we're going to take that and dilute it into smaller containers. So we have these 1.5 milliliter microcentrifuge tubes, and we're going to take a volume out of this starting culture and put it in there. And I'm going to indicate that we have two different things that go in to that first dilution tube. So we're going to fill this up with water, just a simple dilution. Water obviously not containing any cells. And then a portion of this is going to be about 1/10 of the volume, or exactly 1/10 of the volume, so we could do this mathematically. And that's going to contain those cells at the same density in this 1/10, but then when we combine them with the water that's in there by mixing in the vortex machine, then that'll take those concentrated cells and distribute them into the whole volume. So I'm just going to kind of exaggerate this here. Those cells would get distributed so they're not as dense in that next container. So the cells resulting that end up in this tube are 1/10 as many per unit volume. So concentration is 1/10 of what we started. Now the series part of this means that we can do that a few different times. So we can take that resulting solution that we made and we can do the same thing and take 1/10 of that and put it into another tube with water. And as we mix this, then that tube will be even more diluted. So this will be 1/10 as concentrated as the tube that we got it from. I'll call that the previous tube. I'll call the solution. And because this is our second dilution, then we are 1/10 as strong as the solution that was already 1/10 as strong. So we're diluting this 1/10 by 1/10, and that gets us now 1/100th as concentrated as the starting culture. And start. And we use this notation here. We start with the original strength and then we can dilute them 1 to 10, 1 to 100. It's pretty typical to do factors of 10. 1 to 10, 1 to 100, 1 to 1,000, etc. And we refer to these, easier shorthand is to give these scientific notation numbers. So 1/10 is the same as 10 to the minus 1, 1/100th is the same as 10 to the minus 2, 1/1000th 10 to the minus 3, and you can see that's a lot easier way to write these. So we're making basically an exponentially smaller number of cells per unit volume every time we do this dilution. So we can dilute this out, and we'll see the results of plating these onto plates later. So let me just clear this all out. And let's say we take these tubes and put them onto Petri dishes and try to grow those up into cells. So we have our 10 to the minus 1, 10 to the minus 2, and 10 to the minus 3. And let me just kind of exaggerate how many cells would be contained in those tubes respectively. So this first one would be really concentrated, not full strength, but probably more than we'd be able to see. The 10 to the minus 2 would have a smaller amount, and then 10 to the minus 3 even smaller than that. So 10 times less every time we switch over. So if we take the same amount of each of these and spread them onto Petri dishes, then we should be spreading 10 times as many cells on here. Will grow up into 10 times as many colonies. I don't know if my dotting ability is the best here. So probably your 10 to the 1 would be so dense you wouldn't even see colonies, but I don't have time to draw dots over a whole circle. So this would have a lot of cells. This would have also a lot, but 10 times fewer. And this final one would have even fewer than that. So I'm probably not doing a good job of showing you 10 times difference, but fewer and fewer colonies, and eventually one of these would be sort of in a sweet spot of the right number of colonies where you have lots and lots of colonies, but they're separate from each other enough that you can tell that they're distinct. When the colonies are so dense on here, they run together, you don't get to tell them apart from each other.